That looks good to us.
Yes.
Okay.
Perfect.
I think I can manage like that.
Okay.
Okay, so thanks to have me here and to show all the things we did at the FLI for SARS-CoV-2. Today I'd like to give you a short overview of the different SARS-CoV-2 animal experiments we did at the FLI. And please note, it's only a short overview. And for more results, please take a look in the papers. We looked at different farm animals as a potential threat for spillover infections to humans, and also take a look at models for a reservoir host, like vacuum dogs and Egyptian footbeds. And we also evaluate different laboratory animals like ferrets, Syrian goat hamster, and guinea pigs for potential infection models for SARS-CoV-2. So for all these in vivo studies, we used a similar study design. We inoculated our animals with 10 to the 5th TCID50 per animal, intranasal or oculo-oronasal for the chicken. Then, 24 hours later, we introduced three native contact animals to the infectious group to test direct virus transmission. So we take nasal swabs or nasal washing, vector swabs, and also blood samples for testing for viral shedding, and we do necropsy on day 4, 8, and 12 with necropsies on day 4, 8, and 12. Therefore, we have 2 animals of each of the infected group. Because of our study design, we are able to compare all our animal studies very easily. So to make a long story a little bit shorter, we neither get clinical signs in pigs, guinea pigs, or chicken. They showed no virus or show no transmission. We were also not able to detect viral RNA or infectious virus in any of the samples. So we can say that these species are not susceptible for SARS-CoV-2 infection. So let's start with the cattle. They are quite interesting. In the cattle experiments, none of the inoculated calves showed signs of clinical SARS-CoV-2 infections, and no virus transmission was occurred. Here you can see the sample taking in the cattle. In 2 inoculated animals, virus replication, here you can see over here, on left side over here, was detectable at day 2 and 3 in nasal swabs, but the genome loads are very low. The sera of both of these calves were also reactive in the RBD ELISA. In addition, the virus neutralization test showed a visible but incomplete inhibition of virus replication in one of the serum samples taken on day 20 post-infection, seen in the last figure on the left side. So, to sum it up, cattle showed only a slow susceptibility of a SARS-CoV-2 infection, but they are able to replicate the virus. So in the next slides, I will move on to the potential reservoir animal model. We used the exactly same study design for the raccoon dogs and inoculated them with 10 to the 5th TCID50 of the virus. 6 of the 9 infected animals were infected, and viral RNA was measured on day 2 to day 16. You can see over here in the nasal swabs. Virus titer peaks were detected also in the nasal swabs at day 2 here for the titration. We were also able to detect virus in 2 out of 3 contact animals beginning at day 8, but no clinical signs, of course, in there too. Seroconversion was also found in all PCR-positive animals, and neutralization antibodies were observed in 2 of the inoculated raccoon dogs, beginning at day 8, but they were very, very low in activity. So we can also say that raccoon dogs indicate that they might be a reservoir for SARS-CoV-2 or a model for SARS-CoV-2 infection, and could eventually cause zoonotic infections to humans. The next results I'd like to show are from our Egyptian foodbed experiments, and as you can see in the picture at the left side, they really like to stay close together. And so it was no wonder for us to find them all all contact animals get infected, and virus shedding was detected between day 2 and 12 on the left figure. But they also showed no clinical signs. On this figure, you can see the virus distribution and the different tissue samples we take. SARS-CoV-2 genome was detected in the nasal concha. You can see here in the front. In 7 of 9 infected bats, and also in other respiratory tissues. They are the first ones here. Nasal, concha, and also in the trachea and in the lung. We were also able to observe antibodies in all inoculated bats by indirect immunofluorescence assay and by virus neutralization assay, starting at day 8 starting at day 8, and also at 1 of the contact animal at day 21. So all food beds show the food bed showed typical characteristics of a reservoir host. The next few animals I want to present are cute ferrets, and they also show no clinical signs, but the virus shedding was detectable between day 2 and day 12. You can see here. All direct contact animals get infected, and virus isolation was successful from nasal washes collected at day 2 and day 4. As already shown for food beds, the highest virus genome loads and tissue was sampled in nasal conchae. And also we get neutralization antibodies beginning at day 8. And also in 1 contact animal, I see at the bottom here. They start early and with a seroconversion. All these results are collected in this paper published in June 22, and there are also results from histopathology, and yeah, just take a look in there, and I think it's very interesting. At last, I will show you a few slides about our Syrian gold hamster model, and the cute little guys showed white loss up to 20% in our study, but no other clinical signs. We could detect massive virus shedding and found transmission to all contact animals. Here you can see the virus shedding and the genome copies, and they start by day 2, and we have virus shedding up to day 8 for the infected animals, and for the contact animals to day 12. So as I already shown for the food beds or for the raccoon dogs, we also get virus replication in the different tissues we sampled. The most replication we were measured in respiratory organs, but also no respiratory tissues were RNA positive. They also we get also a neutralization antibodies for all animals, also the contact animals. So the Syrian hamster model is a very suitable model for a mild human infection of SARS-CoV-2. So what did we learn from our animal studies? We learned that food beds and raccoon dogs showed characteristics of a reservoir host, and that pig pigs, chicken and guinea pigs are not susceptible for SARS-CoV-2. Ferrets are a very good model for subclinical human infection, and the Syrian gold hamsters are also a suitable model for human SARS-CoV-2 infection. But why are we interested in this? We like to do vaccine studies and do them already with our ferret model in a prime boost challenge vaccination model. After the challenge, we looked for nasal virus shedding, seroconversion, and of course clinical signs or mortality. And for the hamster model, we were interested in antiviral testing of compounds. Therefore, we infected the hamsters, and after normally 24 hours, we applicated our antiviral compounds and looked then for nasal virus shedding, mortality or white loss, and also seroconversion. So about the different antiviral compounds we are looking for, we are using molecular dogging, or our partners using molecular dogging as a model tool to identify new potential anti-corona drugs. We test them in vitro and afterwards we're able to do in vivo studies. We will work with nanobodies, monoclonal antibodies, and of course we are looking for natural compounds and the derivatives to test them against SARS-CoV-2. So at the end, I'd like to thank all the people listed here because they do most of the work, and you for your attention.
Okay, thank you very much, Saskia, for this very nice sum up of the SARS-CoV-2 research done by the FLI. So Sanda, will you take it from here? Saskia, if you can disconnect your present.
I'll look for it.
Okay.
I still couldn't.
You should be able to take over if you press share screen. All right, I think. Yeah.
Looks good.
Can you confirm that you can hear me?
Yes, perfect.
All right, excellent. Magic doesn't always work very efficiently, but it's great that it works now. So I would like to give a bit of an overview of our research efforts on the SARS-CoV-2 virus. And of course, a year ago we were all surprised by the pandemic of SARS-CoV-2. But if you just realize that there's already 4 coronaviruses circulating in humans, and given that we had 2 zoonotic introductions of coronaviruses in humans in the last 2 decades, it's actually not so much of a surprise. And luckily, when the new virus emerged, we could rely on our experiences with the previous coronaviruses. So in 2003, we were part of a team that first characterized the SARS-1 virus in a macaque model as the causative agent of the observed severe acute respiratory syndrome. And later on, we also demonstrated the ability of SARS-1 to replicate and cause disease in cats and ferrets. Then, sorry, I'm struggling now a bit with my slides. Yes, then in 2012, our lab identified the MERS coronavirus from samples from a man with pneumonia in Saudi Arabia. And in follow-up studies, amongst others, we developed an orthopox vaccine that reduced the MERS excretion in dromedary camels. This was actually done in collaboration with the CReSA. And furthermore, we developed a rabbit model for MERS infection, and this model was subsequently used to demonstrate that MERS did not transmit between rabbits via contact and also not via the air. But then a year ago, when the new SARS-2 virus emerged, we started a huge group effort. So all people, well, we already have a coronavirus group, but all people working on influenza, morbidity, arboviruses, they all dropped whatever they were doing. And together with March's people in her new ABSO3 facility, we started to do research on SARS-2. One of the first studies that we actually did, like many others, was to compare the SARS-2 pathogenesis with that of the previously emerging coronaviruses. So therefore, we inoculated some pneumococcus macaques with SARS-CoV-2 and SARS-CoV-1. I just call it SARS-CoV-1 now because it's easier, or with the MERS virus, and we compared the pathology and the virology. In the SARS-CoV-2 infected macaques, the virus was excreted from the nose and the throat in the absence of clinical signs, and the virus was detected in type 1 and type 2 pneumocytes in the lungs. For SARS-1, the lung lesions were typically more severe, whereas they were milder in the MERS-CoV inoculated animals, where the virus was detected mainly in type 2 pneumocytes. So these data, they show that SARS-CoV-2 causes COVID-19-like disease in macaques and provides us with a new model to test preventive and therapeutic strategies. Then the next thing we wanted to do, of course, is when a new pandemic starts, you want to know how these viruses are spread. And we wanted to study if these SARS-CoV-2 viruses were spread either via direct contact, indirect contact, or via the air. And for this, we use the ferret transmission model. So we have already more than 10 years of experience with the ferret transmission model. And in this model, we investigated if SARS-CoV-2 could be transmitted via direct contact, which if you can see my cursor, on the left-hand cages, we have a donor animal and a direct contact animal housed in the same cage. And then on the other side, we have an indirect recipient animals, which is housed 10 centimeters away from the other animals. The animals are separated by these steel grids in the middle, so the only way that this indirect recipient animal can become positive is after transmission via the air. Here we observed that the SARS-CoV-2 virus was transmitted via direct contact in 4 out of 4 pairs and via indirect contact, so via the air, in 3 out of 4 pairs. And this is, by the way, also the ferret model, the ferret transmission model that we offer for transnational access. So we found that SARS-CoV-2 could be transmitted over a short distance at 10 centimeters, which may not be too difficult for a respiratory virus, of course. So therefore, we built a new setup to challenge the virus a bit more. So in this case, we had the donor animal at the bottom and the indirect recipient animal at the top. This model was first validated with H1N1 influenza virus that transmitted in 3 out of 4 pairs that we tested. Then the SARS-CoV-2 virus transmitted in 2 out of 4 pairs. And but actually should have been a negative control in the system was SARS-CoV-1, but this virus actually turned out to be transmitted in 4 out of 4 pairs. We have a lot of explanations for this, but I will not go into all those details now, but I would be happy to discuss that later. And of course, in order to contain the COVID-19 pandemic, it requires to prevent or to reduce the viral transmission between individuals. And this can, of course, be done by vaccines that we are now trying to do or going to try to do, but another option is treatment with compounds that can prevent infection. So we also use the ferret model to evaluate a lipopeptide fusion inhibitor that blocks the critical first step of infection of SARS-CoV-2. So what we did, we had 3 donor animals in gray that we inoculated with SARS-CoV-2. These animals were later co-housed with 4 animals. 2 of them were untreated and 2 of them were treated with the lipopeptides. And these animals were co-housed for 24 hours. And after those 24 hours, those animals were rehoused in the original setting. And of course, we swapped these animals every day, and you can see here that in the donor animals, you can see very nice replication here in gray. Then this gray area shows the 24 hours that the animals were co-housed. And you can see that after co-housing with untreated animals, you can see that the untreated animals also become positive, as indicated by the red symbols and the red line here. But you also see that the animals that were treated intranasally with the lipopeptide, they did not become infected because there was no virus shedding in these animals. So those lipopeptides could prevent infection, although there was 24 hours of exposure to an infected donor. Next, we also explored the hamster as a model for SARS-CoV-2. So these animals turned out to be sensitive to infection with SARS-2. There was a weight loss of approximately 15%, and replication in the upper and lower respiratory tract was demonstrated, as shown here by the nasal, the shedding in the nasal washes, and you can see here the pathology and the lesions in the lungs. So in addition to vaccines and other treatment options that prevent transmission, like I said before, effective clinical intervention strategies for COVID-19 are also urgently needed. And several clinical trials have evaluated the use of convalescent plasma containing virus neutralizing antibodies. However, the effectiveness has not been proven yet. So it's not shown on this slide, but we demonstrated in the hamster model that when we treated hamsters with a high dose of human convalescent plasma or a monoclonal antibody, that these animals were protected against weight loss, and it showed reduced pneumonia and pulmonary virus replication as compared to the non-treated control animals. And however, a 10-fold lower dose of convalescent plasma showed no protective effect. So this was a very interesting observation that for variable relatively low levels of virus neutralizing antibodies in convalescent plasma may limit their use for effective antiviral therapy, which favors the concentration of the use of concentrated purified monoclonal antibodies. All right, I went a bit rapid and I have no conclusions slide. The idea was just to show you, to present you with an overview of the efforts that we made. And I only presented here the work that we did to develop animal models and to show you how we use the animal models to see if which treatment options can work for SARS-2. So this is a whole list of people that contributed to the studies. It's not even everyone who helped. And of course, this research was, among others, sponsored by VetBioNet. And with that, I'm at the end of my presentation.
Okay, thank you very much, Sander. Very impressive piece of work. There's one thing I forgot to state earlier. The questions we will have collectively at the end of the session. Okay, I think it's more sensible that we share the questions with all presenters. Okay, so we switch to Nora then. Your microphone is still muted, Nora.
I just remembered. Thank you.
Okay.
But now you can see my presentation, you can hear me, right?
No, we don't see your presentation anymore.
All right. And then let's do this once again. Now it's back?
Yep.
Yes.
Yeah, so thanks also giving us the opportunity to briefly present what we have been doing on SARS-CoV-2. I'm presenting here as part of a larger team with many more scientists involved at our institute. We had a partnership with CEPI already existing for Rift Valley virus infections. And CEPI, you've probably heard of it. It's a coalition for epidemic preparedness and innovations. It's an international coalition to be better prepared in case a pandemic or epidemic situation occurs in humans, and that is exactly what happened this year. And as a part of this partnership, we started investigating several different animals, whether they are susceptible for SARS-CoV-2 infection or not. We were looking into mice, where we looked at 3 different types of mice, aged mice, diabetic mice, and obese mice, as part of to mimic the risk factors that we also see in human patients. We were looking into pigs and into Syrian hamsters. We also performed 2 studies in domestic cats, and our institute is also quite busy studying minks, but not as experimental animals, but more in terms of field infections. And I will give a very brief overview of all of these studies that we have been doing. I will start with the mice. For mice, we didn't see any clinical signs we did observe very little body weight loss in all of the animals, but it could be related only to anesthesia because they recovered quite quickly from the body weight loss. We didn't find any macroscopic pathology, so there was no pneumonia present in the mice. However, we were able to stain SARS-CoV-2 virus in bronchi epithelium. We didn't see it at all in the alveoli. Interestingly, we also collected throat swabs from the mice, and they stayed positive up until day 10 post-infection. And we were also looking for replicating virus by performing a subgenomic PCR. And also there, we did detect positive signals. So virus was able to replicate in these, in few of these mice, not all of them. And then we analyzed these positive lungs and throat swabs from these mice by deep sequencing, and we actually found that there were that mutations were occurred in these strains. So I think the virus is very rapidly adapting to a new host if necessary. And yeah, in all of these cases where we found a replicating virus, we did detect very similar mutations. I was asked to focus a bit more on the pigs. So here I'm showing a very brief overview of the experiment. We had temperature transponders that we transplanted into the abdomen. We infected pigs by aerosol and by intratracheal infection. We performed euthanasia on different time points post-infection and collected all kinds of samples throughout the study. It's a quite similar setup as also FLI has done, and I think most other people are doing just without contact animals in this case. Pigs, in our case, didn't show any clinical symptoms, no fever, no body weight loss, quite comparable to what also all the other people have seen. So they seem to be quite unaffected by the infection. Also, we couldn't find any virus shedding in any of the swabs. We collected nose swabs, throat swabs, and rectal swabs. And only up until day 2 post-infection, we found some viral RNA, which is most likely only the challenge inoculum. Interestingly, however, we did find genetic material up until day 10 post-infection in the lungs. But we also looked whether this would be replicating virus or whether subgenomic PCR would be positive, but it wasn't. So we think that this is simply remaining challenge inoculum that we find in these lungs of pigs without it leading to an actual infection. We euthanized pigs post-infection, so we did not have the same number of animals at all the different time points. But interestingly, towards the end of our study, which was 3 weeks, we did find that some of the pigs actually have developed neutralizing antibodies. So on day 10, we had 4 pigs in total, and 2 of these 4 pigs had antibodies. And then of these 2 positive pigs, 1 went to necropsy on day 10. So then on day 14, we still had 1 positive pig with positive with neutralizing antibodies. And we, the lungs were also quite clean, so we did not find any pathology in them. And we find this quite interesting that, pigs apparently are able to make antibodies post-infection, even though there is no active virus replication. We performed also 2 studies in cats. First, a pathogenesis study, and thereafter a transmission study. All of our infected cats became positive in all kinds of swabs, nose swabs, throat swabs, mouth swabs, rectal swabs. I'm just showing here some nasal swabs up until, let's say, one week after infection. And all of these infected cats also developed antibodies, approximately at the time where viral titers drop in the swabs, antibodies start arising. So that is those are results from the pathogenesis study. We also looked on how virus is shed so it can be detected in environmental samples, so from the feeding tray, from the litter box, from the toys that they have, and indeed we do find viral RNA on all of these samples, and even in the air. So in the air in our animal unit, we could also detect the virus. And this led then to the second study, the transmission study, where we're currently analyzing the data. There we had an infected cat and a direct contact cat and 2 indirect contact cats, where we want to see whether they have been infected only via the contaminated environment. And then I also want to share some data from our hamster model. We have a so-called activity cages that we use in our study. So that's it's a running wheel that the hamsters have, and that one is connected to a counter that with a magnet which counts the rotation of the wheel, and that is a measure for the activity of these hamsters. And what you can see in the upper graph is that a post-infection activity drops to almost zero, starting from approximately 10 kilometers a day that they are running to almost nothing, and then recovering from that. Similar to also what the other groups are seeing, body weight loss is about between 10 to 20%. And what we see in our model is occasionally respiratory disease. So we do have a couple of hamsters that really have difficulties breathing. And this led us to purchasing a whole body plethysmograph, also with the funding from CEPI. Which is a device how you can measure the respiratory function of small animals. Let's say the breathing frequency, the tidal volume, the capacity, and so on. And we have been using that for a couple of studies now, and it seems to be able to pick up worsening of the disease. For hamsters, we find positive virus positive swabs, both for, let's say, the
Nora, are you still there? It seems. Can you hear us, Nora?
The voice lost.
Yes. She's still shown in the participant list, so she hasn't quit.
She has the micro out.
Yeah, and the camera also. I mean, it disappeared. I have all the bioactive cameras activated and she's not there anymore.
She was offline, but I think she just connected back.
Yeah, it seems. I can see her cursor moving.
Yes, we can see your screen, your cursor, but you will have to reactivate your microphone and camera.
Looks as if she would continue.
Yeah, it's possible that she she didn't hear us at all. Norbert, she's not by chance just in an office next to you or something?
She's in the next building, but
Okay.
I will walk to her. I will. But I think she's already done.
Yeah. Okay. I haven't carefully followed. I think she was presenting the mink study at the end.
Yes.
Perhaps we will switch to Julia now. Meanwhile, perhaps you can try to call her, Norbert, and ask that she reconnects and that she can perhaps show the last slides again. Okay.
I will do so. Hopefully, Julia can already. Yeah. Okay.
Yeah. Can you see it?
Yeah,
so?
yeah, we can
Yes.
see, we can hear you. Everything is perfect.
Okay, let me put, because it's not in the presentation mode, right?
No.
But I cannot select. Let me check this because now. Okay. Now?
Excellent.
Okay.
That's great, yeah.
So first of all, thank you very much to organize this meeting, because I know and I can imagine that it's not the same to organize a non-virtual event. So it can be even more complicated to organize an online event. So thank you very much. And also it's a pleasure to be here presenting the work that it has been done related to COVID at IRTA-CReSA, together with our colleagues from FLI, EMC, and Wageningen. It's a really pleasure. So I will try to summarize a little bit. As they also said, we have been involved in several type of work, and I try to summarize, but I'm not sure if everything will be clear. So at the end, I will be open for questions, of course. No, it's not changing now. So, well, as probably you all know, CReSA has BSL2 labs, but of course also BSL3, and what is more important, let's say, for our work, the animal facilities inside the BSL3. Well, for those who doesn't know, IRTA-CReSA is located approximately 20-25 kilometers far from Barcelona, so in the surroundings. And then, yeah, the biggest part, let's say, of the BSL3 is the animal facilities that we have 10 boxes for large animals, and we have been performing experiments with swine, poultry, and other animal species, ruminants, horses, camelid species. This, of course, mainly for the MERS coronavirus, and other, also some wild goats and other animals. And then we have 2 boxes for rodents. Mainly, we are working with mice, rats, and hamsters. And also because of the COVID situation, and even before, we were thinking that we need more space for rodents. So in principle, in the coming year, we will have an extra box to perform rodent experiments also. So what we are doing, what we did at the beginning, so of course what we first needed to have was the virus, and nowadays we have some local isolates, but also from international, from the US, and also from Berlin. And then we set up some techniques, of course, virus isolation, virus titration, and then quantitative PCR, both genomic and subgenomic PCRs, some immunological assays, such as neutralization, also ELISAs, of course, and then some pathological assays, of course, histopathology and immunohistochemistry. And then I want to summarize also a little bit the in vitro and in vivo models that we have been developing and using in our studies. So I will start with the in vitro work. As also some of my colleagues have mentioned, they were studying some antivirals. We were also interested in studying some of the existing antivirals or drugs against COVID. And then we did some studies mainly in virus cells, but we also have CALU models and then other cells that we are using together with our colleagues at IRTA-CReSA. These cells we are using to test the antiviral activity, as I told you, of several drugs, and we are performing both assays with pseudoviruses, but also with the live virus, of course. And then the assay we are using is checking the cytopathic effect by a luciferase assay. And then here on the top, you can see, well, we submitted our article. Now it's in this public platform, let's say, but we are trying to submit also in a journal. And then, of course, we also as we are working together with hospitals, we are performing several seroneutralization assays with plasma from COVID-19 patients. And from all these studies, we have been also publishing several works. The main findings that we found is that stable neutralizing antibodies have been shown in patients at least until 6 months after a mild and severe episode of the disease. Of course, and mainly our doctor colleagues, they are also studying the cellular immunity and the role of cellular immunity. And then before this, we also detect this increasing of neutralizing activity. If you see on the left side of the screen, here the patients were divided. So if they were asymptomatic or mild symptoms, until if they needed the ICU, if they were in the ICU, and this correlates with the activity or with the high neutralizing activity. So these were some of the in vitro work that we were doing, because we are also performing some studies for company, not only to test antivirals, but also to test some antiseptics and also some devices, big devices to inactivate the virus. And then, importantly, we are also doing in vivo work, of course. We also perform a study with pigs, and the same as Nora said, we didn't find evidence of infection, but we also detect this immunogenicity in the animals, because here we inoculate the animals. We did 4 different groups, one intranasally inoculated, the other one's intratracheally inoculated, and then one via intravenously and one group intramuscularly. In these both groups, intramuscularly and intravenously, we detect antibodies against the infection. And this is already published. And then we are now focused and mainly working with transgenic mice and golden Syrian hamster. We also did a small trial with aged mice, but we couldn't detect, well, they were not susceptible to the infection. So for the moment, we stay with the human AC2 transgenic mice and the golden Syrian hamster. This work, well, Kim Segales and myself, we are part of the COVID animal model of the WHO working group, together with some also of the investigators here from the VetBioNet. And then we agree that these 2 models are really good to study antivirals, antibodies activity, and also vaccines, And we believe that golden Syrian hamster recapitulates better the disease in humans. So we really think that this is a good model. And then from both species, of course, first we needed to set up the animal model. And then with transgenic mice, we did some experiments testing antiviral efficacy, different antivirals well, mainly one that worked really good in vitro. So the company asked us to do an in vivo model, and hopefully the results will come up really soon. And then with golden Syrian hamsters, well, the test of antiviral efficacy will come in the next year, but we did a study to assess the reinfection, and we have the manuscript on preparation. And then for the upcoming year, we have already 4 studies planned to do vaccination studies with this model that we already set up. And then this is another story. Let's say we also have a project to detect SARS coronavirus in pets and also in zoo animals, let's say. And in fact, the story began last April when we detect the first cat in Spain positive, and it was a cat owned by a COVID-19 patient. So from here, we obtain a project, and what we are doing is to test cats, dogs, and ferrets. And at the moment, we didn't detect by PCR any other animal positive from all the ones that have been tested. Now we have the sera, and we will do also, of course, serology. And maybe from here we will have some positive from the serology, but not from the virus. And then recently we have not yet published, well, published in the ProMed, but that the 4 lions at Barcelona Zoo, they turned positive for COVID-19, and now we are extending this study. In fact, we follow the complete kinetics of the infection of these animals, and we will try to sequence also the virus, and also to do the serological study of these animals. And well, all this work started with a huge collaboration between, and this is really a good example of the One Health principle, because IRTA-CReSA, no, we are the animal side, but we are collaborating with Barcelona Supercomputing Center. They are the bioinformaticians, and with IRTA-CReSA and the Hospital Institute Germans Trias i Pujol, that they are the medical side, let's say. And all this big consortium, the CIVIC consortium, is supported by Grifols, a pharmaceutical company, but also from the national, so the Generalitat de Catalunya, and by a crowdfunding, Jaume Corono. So we started from March, April, we could start doing all this research. And then, of course, we are also collaborating with other research groups and industrial partners. We are offering animal models as well as these in vitro models I was briefly explaining. And last but not least, we also continue our work on MERS coronavirus. And in fact, last Friday, our PhD student, now Dr. Teniger, he defended his PhD thesis, and the thesis was mainly, so the main objective of the thesis was to study the early immune responses against MERS coronavirus in camelid models. He was mainly working with alpacas. And then all this COVID situation was bringing more people at our lab. So besides Jordi, Kim, Albert, Teniger, and myself, now we have Marco, Cristina, Nuria, and all other people from CReSA that have been involved at least at some point, and of course they are really important to perform all this huge amount of work that we have been involved in. And that's it from my side. I try to summarize all the work, And if you have questions, I think now it's the turn.
Thank you very much, Julia. Very nice summary. Plenty of fantastic and very diverse studies. Really, really great. Nora, you're connected now. Can you hear us?
Yes, I'm back. I don't know what happened. I think my go-to meeting crashed.
Okay. Perhaps you can, before we open the question round, I think we have only missed your slide concerning the mink study. If you would just like, or just share the slide, or just talk briefly about it, whatever you prefer.
Yes. So let me see if I can share it without it crashing again. So basically, what we have seen in the minks was mostly those were field infections. So the animals there presented with gastrointestinal and respiratory symptoms, and we performed many necropsies on these animals. And you can see here on the macroscopic slide that that's quite a severe interstitial pneumonia. The lungs are quite affected. And you also see that here on the histological slide. Obviously, these animals were not exsanguinated, so that you still have a lot of blood in the lungs, but also the tissue is really super condensed and highly inflamed. I wanted to say also that from all the sick ferrets, only 1 to 5% die in the end. So that's not as lethal as sometimes it is said in the news. However, it is different than in other animal species that we have been investigating. So the animals actually die. And what we also saw is that the initial introduction into the mink farms is likely from infected human workers there. So it was a transmission from humans to minks, but we also found then transmission from minks back to humans. So it's really a zoonotic cycle that is fulfilled in these animals. Yeah, that's what I wanted to say about that. And that, yeah, I think that even though those are field infections and it's not an experimental model in our institute, at least, those are still quite relevant animals to keep in mind for SARS-CoV-2.
Okay, thank you, Nora. So then we have a couple of minutes for questions. That's great. And I have already one question here in the chat. There was another question earlier. I will start with the question that Martja is asking. So Martja asks, how do in vitro models predict animal host specificity and or antibody or drug therapeutic efficacy? I think at least 3 of you have worked also, or several of you have worked with in vitro models. Julia, you want to?
Yeah, this is a very interesting and nice question, because of course, at least for us, when I think it was in April, May, when we did the experiment in pigs, we of course did the experiment. It was not a blind experiment, let's say. We had some bioinformatic predictions, but also some evidence from some other guys that said that pig cells at least may be susceptible to infection. So that's true that maybe it's not 100% the prediction of a specificity when you are working in vitro with in vitro models, and then you go, for example, to the in vivo model. Of course, it's not the same. It's not the complete organism. That's true. And concerning the antibody and drug therapeutic efficacy, we have also seen some reports showing that maybe some antiviral is really nice working in vitro, but then you go to the in vivo model or in humans and doesn't work. To our experience, the one that I was saying, it's an antiviral that in vitro work even better than remdesivir. and in vivo we also get good results, let's say. So I think that in vitro models, we cannot skip them, because when you have a huge panel of candidates to test, so you at least can do a screening and you can have an idea. But then, of course, that doesn't mean that the results will be translated 100% in an in vivo model. And then, of course, I think also we all know, no, that sometimes even in vivo works and then not in humans. So that's true. I think it's really important, or an important tool to screen, and also it's really important to know what we want to test, because we have been testing also some candidates that they work, or we can see their activity, for example, in CALU 3 cells, but not in Vero, because they are anti-inflammatory compounds. So it's really, yeah, the question is really important, and I think we really need to focus on what we want to answer. But that's true. I totally agree with Marcia.
Okay. There's another question from Mathieu Epardot here from our INRA-ISP center. He has already asked during the session if he can ask a question. So his question is, Saskia mentioned neutralizing antibodies in hamster against spike only, where systemic, nasal, wash. Al, did you check for IgAs, neutralizing or not?
It's a good question, but we did. I didn't do that work, and we didn't start this work now. We have a cooperation partner who wants to check neutralization antibodies in hamsters, but we didn't do anything about that now.
Okay. Then I think Ryan Waters from Burbright Institute has a question also. Perhaps Ryan, it's the easiest if you connect and ask your question.
Okay, yeah, thanks very much, Sasha. Yeah, no, great set of presentations. One thing that just struck me that is there an agreed approach for defining replication? So, for example, you know, a set subgenomic RNA approach for PCR analysis or assessment of replication, and or non-structural protein ELISAs, and or, you know, protein localization in tissues. You know, has there been an attempt to sort of define a standard set of approaches for defining what replication means?
So that's true that, for example, we are using the subgenomic RNA technique, and we can say no that here there is a kind of replication, but to us at least let's say the golden technique to say replication is to check the infectious virus. That means in Vero cells to titrate, so we can have a genomic PCR, a subgenomic PCR that it's more precise. Let's say, but to us replication means to isolate the virus in swabs, in tissues, in whatever sample.
Yeah, but we for instance we have we're not successful to isolate virus from swabs, but they are all positive on the subgenomic PCR. So we define this differently. So we say that we find replicating virus also in the throat swabs, and that we cannot isolate virus from these samples.
Yeah, when we talk about subgenomic RNA, we are also talking about replicating virus, but to see, you know, if it's replicating on cells. But that's true, and we are also having some problems, even with low CT values, or that it will be expectable to have virus isolate, and that's true that we also have. So that, let's say that the limit of detection or of isolating virus, it's really low in this case. Yeah, that's true.
Yeah, and they're different. They're like, you can have genetically modified Vero cells, which are a bit more, let's say, easier to be infected. But then, yeah, you're comparing also again different systems.
I have a question for Sander. I'm curious now after your presentation. So why do you think SARS-CoV-1 is more readily transmitted by airborne infection than SARS-CoV-2?
Yes, it's very interesting. We've been thinking about it for a long time because, like I said, it was a bit our negative control because we did not expect any transmission there. However, if you then look, so we looked at the differences in SARS-CoV-1 and 2 replication in humans, where in humans you can see for SARS-1, the peak of replication is only in the 2nd week, so after 10 days, usually when people are also already very sick. So and also in the beginning, the first phase of infection is mainly a lower respiratory tract infection and not so much upper respiratory tract infection in humans. So on one hand, we think that we did not see a lot of airborne transmission in humans because on the first period, there was a lower respiratory tract infection. When it was more in the upper respiratory tract, then the people were usually already hospitalized and not walking around in the streets anymore, which may explain why SARS-CoV-1 did not transmit too much in humans, and maybe that's the reason why at that time it could be, the outbreak was stopped. On the other hand, we also think that ferrets are extremely susceptible for infection with coronaviruses, so SARS-1 and SARS-2. That's also backed up a little bit by what you can see happening in the mink farms, right? When the virus really spreads very fast through the farms, that is also can be via contact, but also via air. So if I understood those outbreaks in farms can be really patchy, so it's not that it goes from one side of the farm to the other side, but it can really have local patches of infected animals, which may suggest that it can also be transmitted via the air. So I think it's a combination of different replication and disease in humans and the sensitivity of ferrets, because also in our model, the donor ferrets are infected with 10 to the 6th virus particles in the upper respiratory tract. So if it just replicates there a little bit, maybe it's already easier to be transmitted to a neighboring ferret. I hope it answers a bit your question.
Yes.
At least it's our hypothesis for now. Yes.
Are there any more questions?
Yeah, may I ask a question? We have heard very nice presentations on various and very often the same animal species in various institutes, and we have now a history of about 6 months of animal modeling, and I'm wondering whether there is now some consent on the model to use for various purposes, and whether we are already in the status that we are quite consistent in the use of the model.
If I may, Norbert, the drawback on the hamster model, which is a nice pathological model, is that there is really little information about pharmacokinetics, etc. in that species. Also, there is not a full panel of immune response reagents. So we overlook the hamster as a model. We know a lot more about mice and rats. And that means that not every compound interesting to be tested for efficacy has enough information with it regarding the route of administration and the dose, etc.
I totally agree, Martin, and that's true. The only problem with the mice is that at day 6, 7, they suffer really from neurological signs, clinical signs. So of course, if an antiviral can overcome this is great, but let's say this is not really the natural or the similar disease as in humans. So that's the reason. But of course, I agree, and we have these problems when we want to assess, for example, to go to more immunological aspects. We did experiments, for example, with Luminex, and this we did it in mice, with mice. So some experiments we cannot do it in hamsters, or we need to to look for reagents or maybe to develop new reagents. And yeah, I totally agree with this.
Okay, is there any more question? I would love continue discussing with you, but I think we will close the COVID session then for now. Thank you very much to the presenters. It was great. Thanks a lot. Very instructive, very insightful. And so we will have another coffee break now, I think. Let me check the agenda.
Yes.
That should